Okay, I'm Rima Kabaz, and it's a pleasure to moderate this morning's session. Our first speaker this morning is Dr. Gerald Tuskan, who is group leader of the Plant Systems Biology Group at the Oak Ridge National Lab. Dr. Tuskan is a graduate of Texas A&M, and he has one of the top cited papers in science over the past 5 years for the work where he led the sequencing, assembly, and annotation of the Populus genome. It is not just the one top cited papers, but has been cited more than 1,500 times. Very, very impressive. Very prolific in publications, invention, disclosure, and patent applications. And so we're delighted to have him discuss with us this morning plant-microbe interactions in the Populus rhizosphere.
Well, thank you for that introduction, and it's a pleasure to be here. I'm looking forward to all the talks. I'm here today to tell you about the most diverse microbiome on the planet, and that occurs in dirt. And although we don't think of the environment as very forgiving in terms of what it can contain, in a single tablespoon of soil, the microbial diversity exceeds the entire human microbiome by orders of magnitude. And so we're talking about a very diverse ecosystem, and that is controlled in part by the environment, the temperature, the moisture, but also by the host genotype. Sure. Little adjustment here. Is that better? I'll cover a little bit of background about the relevance of why we're looking at the popular microbiome, tell you a little bit about Populus, a little bit about the soil environment, and then explain or describe some of the results we've gotten to date. This effort is a large collaborative effort funded by the U.S. Department of Energy, and I'm going to highlight results from a group of scientists in my group. Populus is an early successional tree species, so it's a woody perennial plant. It's shade intolerant. It invades open environments. It's among the largest organisms on the planet. It clonally propagates itself in large clonal stands. This is aspen in Colorado. That's a single genotype that occupies about 125 acres. It's about 5,000 years old. So if you think about this organism, this single genotype cast across the landscape over that period of time, it's faced many climatic changes over the course of its life cycle. And it's accumulated bacteria and fungi within its rhizosphere and in the root system itself. Populus is dioecious. It has male and female genets. That's very unusual among plants. And its poplar trees are one of the most polymorphic organisms on the planet. The genome was sequenced about 5 years ago. It's about 485 megabases, and there are 41,000, 42,000 genes in the genome. It can be easily transformed, so we can genetically manipulate it. We can introduce RNAi constructs to knock out gene expression. And we've used all of these tools to advance our understanding of the microbiome. The project is funded by the U.S. Department of Energy. I wanted to talk just a little bit about applications. So cellulose and lignin are the two most abundant polymers on the planet. Cellulose makes up paper and wood, the tables, the chairs we're using here in this room, the paneling on the wall. Lignin is the glue that holds cellulose together. If you can deconstruct the cell walls, which are made up of these two polymers, you can use the carbohydrate form in cellulose and ferment it into transportation fuels. You can take the lignin and spin it into carbon fibers. So the U.S. Department of Energy is interested in producing plant materials that can displace transportation fuels and make high-value, low-weight materials for aircraft and automobiles. Another application the U.S. Department of Energy is interested in is carbon sequestration. We've all heard about global climate change and elevated CO2. Most of the carbon that's stored in the planet is stored in soils, and most of that carbon comes through the process of photosynthesis, captured out of the atmosphere in the form of CO2, converted into glucose, and then that glucose is converted into cellulose and lignin. If we can increase the amount of carbon that comes in form of lignin below ground, we can increase residence time and reduce the impacts of elevated CO2. So all of these applications are important to the U.S. Department of Energy, but it became apparent about 10 years ago that it wasn't just about the host plant. So we can modify or manipulate cellulose content, we can modify lignin content, which has impacts on products or ecological function. But when we deployed those genotypes into an open environment, they didn't behave as they did in the greenhouse or in the laboratory. And what was the difference was the microbiome. So we began studying the diversity that occurs in the soil system around the roots of these plants to try to characterize how the microbiome changes product, how it changes the cell wall composition and ultimately the product output. This is the approach to the project. We've been funded for 3 years. Basically, we isolate individual communities from the root system, the rhizosphere of a poplar tree. We isolate these organisms, place them in culture, and have begun looking at the signaling that occurs between the host and the microbiome. I didn't mention earlier, but I should mention that the microbiome is composed of not only bacteria, but fungi and archaea. And so we're looking at all 3 taxa. And we want to understand the communication, the chemical signaling that occurs between the bacteria, the fungi, and the host, to see if we can then ultimately reconstruct the community in a predictive manner. I talked about the diversity that occurs in soil, but there's a fundamental ecological relationship that's not very well defined. And that's the relationship between diversity and function. So hypothetically on the red line, as diversity goes up, does function continue to increase? Or alternatively, as diversity goes up, does function plateau? And so we're working under what's called the lottery hypothesis. You can think about this as a shopper in a bazaar, an open market anywhere in the world. And there may be many, many kiosks that sell similar things: clothing, food, pottery, lighting. And so as you enter the open market and you begin purchasing or acquiring these products or these goods and services, you don't always go to every store or every kiosk that offers spices, for example, or lighting or clothing. Typically, a shopper will pick the first or second that they find. And so we're applying that economic model to how a plant recruits its microbiome as a testable hypothesis. And so we want to define this relationship. We want to understand how plants acquire their microbiome. This is the conceptual approach. We've sampled field sites over 2 different years in 3 different environments. We've looked at native collections of poplar in North Carolina and Tennessee, and we've looked at common garden. We've isolated thousands and thousands of microbes from these environments. We've characterized them under resequencing and pyrosequencing. We've taken measures of function. We're looking at chemotaxis, quorum sensing, biofilm formation, and looking at signaling between the fungi, the bacteria, and the host, and trying to determine ultimately the composition here so we can recreate the community. Give you a little background on the environments that we've sampled. So we've sampled along 2 river systems, one in North Carolina and one in Tennessee, and we've collected microbiomes over multiple seasons, in multiple years, over multiple genotypes. This gives us the ability then to segregate or separate out diversity at the pyrosequencing or OTU level in terms of abundance and composition of the communities, and determine which of the environmental factors, whether it's location or host genotype or season or year, that controls diversity. We've also collected 1,000 individual genotypes of Populus along the Pacific Northwest. Those individuals came from native trees that existed from central British Columbia down to California, which is off the map. They represent changes in native environments for moisture, pH, temperature, photoperiod, elevation. And since these are native trees that have existed on the environment for tens of thousands of years, when we bring these genotypes into a common garden, we're able to measure the effects of that genotype on microbial diversity. So all 1,100 individuals have been resequenced to a 30x depth, excuse me, and they've been clonally replicated in 4 common gardens. Each common garden has 3 to 4 replicates, so we have 12 to 14 observations per genotype, per phenotype. So from those environments, we've done basic pyrosequencing, and we've looked at diversity in the rhizosphere and in the endosphere. The endosphere is the area of space intercellularly between the individual tissues of the root system. We find a wide variety of diversity in the microbiome, both in the rhizosphere and in the endosphere. The endosphere turns out to be a subcomponent of the rhizosphere. So it looks like host genotype is selectively allowing certain microbes, both fungi and bacteria, to enter its cell system and tissue system. The biggest difference, and I'm sorry, a bit of this is out of focus, but the main points are that the biggest difference between the total diversity we see here is not only just the year, but the difference between endosphere and rhizosphere. And so you see compositional differences that equate to that. The second largest factor is host genotype. And so once you've separated endosphere and rhizospheric differences at a multivariate level, the second largest driver to diversity abundance is host genotype. And the least affected or least impactful variable is year and season. And so when we went back to the same trees and the same root systems over multiple seasons, over multiple years, we isolated basically the same microbiome, both from the rhizosphere and the endosphere, which means that the community is relatively stable over a 3-year period. We were also able to isolate about 2,800 individual taxa from the rhizosphere, and we did pyrosequencing, and we were able to demonstrate that we were able to recreate a representative collection of individual bacteria from the soil, the soil in contact, physical contact with the root system, and from the endosphere. And they're made up mostly of pseudomonads. And so we've reconstructed, we took individual pseudomonads and sequenced those genomes and created a phylogenetic tree. Although at the OTU level, at the pyrosequencing level, all the pseudomonads clustered together, when we individually sequenced and reassembled those genomes, we found a great abundance of diversity. So the core genome is about 3,000 genes in the pseudomonad cluster or clade that we have here. The pangenome is about 11,000. Individual representatives of each of these clusters carried with them individual traits, individual genes that we believe influence their ability to colonize the endosphere of the host. And some of those differences relate to nitrification, phenanine production, and growth-promoting quorum-sensing molecules. We established a quorum-sensing assay. Quorum sensing was not predicted to be a large component of the ecological system that Poplar exists in. Quorum sensing was abundant in the pseudomonads. About 84% of the alpha bacteria ended up having quorum sensing machinery, not only the receptors, but also the signaling machinery. One of the novel discoveries was that in addition to the standard bacterial quorum sensing machinery, there appears to be a sensing pathway found in several of the isolates, about 137 of the isolates, that are receptive to coumarate, which is a polyphenolic ring that's produced by the host. And so the host plant exudes coumarate in the form of this polyphenolic, and then that polyphenolic acts as a clustering signal for the bacterial community. It varies by isolate, and it varies by location. We've also evaluated the biofilm forming capabilities of the microbiome here, isolated from the root systems that I talked about. And biofilm formation varies greatly among isolates, as you might imagine. I won't talk more about this right now, but in a moment I'll mention it again. In addition to the bacteria, we've also isolated fungi from the rhizosphere and endosphere, and they vary. We've taken this fungal diversity and re-inoculated Populus deltoides with the soil as an inoculum, and we've inoculated 2 alternate species, Populus tavia, which is loblolly pine, and white oak, Quercus alba. It turns out that a portion of the poplar endospheric fungal community can colonize alternate species. So they're not host-specific, they're not genus-specific, but rather generalist in how they behave in the environment. Many of these have been isolated and have been sequenced. Again, you can't see the taxa here, but just note that there's wide phylogenetic diversity and that we've sampled many representatives of this community and have sequenced those genomes as well. There are 208 new fungal genomes. And the interesting thing was that although we obtained the soil from Populus deltoides and inoculated white oak and loblolly pine, it turns out that the endophytic fungi appear to be more unique to cross-species colonization than the rhizospheric community. And so it appears again, same story that we saw in bacteria, was that the poplar host genotype selects its endophytic community from the rhizospheric community. Now we've begun doing combinatorial experiments. Here we're looking at mycorrhizal helper bacteria. Mycorrhizae is a form of fungi that colonize plant root systems. It turns out that mycorrhizal fungi probably allowed plants to colonize terrestrial environments. So as aquatic plants moved out of ocean or lake environments, they acquired the ability to symbiotically form relationships with mycorrhizal fungi. We've taken the set of 280 fungal isolates and in a pairwise combination inoculated those fungal isolates with alternate bacteria. It turns out that some of those bacteria have a negative effect on hyphal growth, colony diameter, and survival of individual fungi, and some have a beneficial effect. Again, this is just showing the data across the number of bacterial isolates and the differences in both colony diameter and apex number, which is a measure of branching of the individual fungi. When we take those helper bacteria in combination with the mycorrhizal fungi and we inoculate the host, we see positive differential effects on the host. So it's not a simple one organism to one organism pairwise response. There appears to be a combinatorial interaction between the bacteria, the fungi, and then ultimately the host genotype. And that's demonstrated here looking at secondary root number in a single Populus clone, D124. You can see that here, and also the same effect on root length. And so D124 is the poplar host, GM41 is the bacterial associate, and S23AN is the fungal isolate. And you can see that there's combinatorial benefits of having both the bacteria present and the fungus present, but there's an additive effect, a synergistic effect when you combine all three. Looking at the host side, we've done QTL analysis and association genetics. I mentioned earlier that each of the 1,100 individuals have been resequenced. We've identified regions of the host genome that control Mycorrhiza colonization. There are many genes in these QTL intervals. And as a matter of fact, 74 candidate genes. Several of them are in known plant pathogen-associated gene families. One of them was D-mannoslectin binding protein, which occur in these large tandem repeats. We used association genetics to identify individual SNPs at the gene level. It turns out that those alleles at the D-mannoslectin loci that are beneficially controlling Mycorrhiza colonization have a poly-Q tandem repeat. And so we've verified that this occurs across the native populations. We've identified this within the association study, went back to the native river systems, did qPCR, and verified that the poly-Q allele is the allele determining the favorable interaction. We're now in the process of taking individual host genotypes that have varying amounts of signaling, both in forms of their metabolites as well as small secreted proteins, and putting them in combination with alternate bacteria and alternate fungi. As you can imagine, when you have a collection as large as 2,800 isolated bacteria, 280 isolated fungi, that pairwise and tertiary combinations become outlandishly large. And so we've begun doing genetic selection at the bacterial level in a predictive manner, trying to determine which combinations of bacteria and which combinations of fungi represent the most genetically diverse combinations. Getting back to the lottery hypothesis that I mentioned a moment ago, it may not matter what the taxa is. What may be more important to extend the hypothesis is the genes that come with that particular taxa. So independent of phylogenetic position, we're taking an assessment of genetic diversity in the bacterial and fungal communities and isolating a subset of those, 200 bacterial isolates and about 20 fungal isolates. From those 200 bacterial isolates, we're running a series of functional screens, and then those functional screens will allow us to predict combinatorial representative communities that we will inoculate alternate host genotypes. So this is one of several experiments that we've done where we've recreated a constructed community. We've taken 5 phylogenetically diverse bacterial isolates that each bring an alternate set of functional types. We inoculated the popular host, both trichocarp and deltoides, and observed growth over a 21-day period. You see a substantial increase in growth versus the control over the 21 days versus the inoculated, both in trichocarp and deltoides. So we see a favorable enhancement in growth, which we had predicted based on functional assays, quorum sensing, biofilm formation, chemotaxis, as well as the host genotype, where deltoides, I forgot to mention, initially puts up a metabolic defense against bacterial colonization. But when favorable bacteria are present, the metabolic network is subdued. And so here we have a rapid response genotype. Here we have a neutral genotype to bacterial colonization. So we predicted that these would have favorable effects on both the neutral and the negative responding genotypes. In fact, they did. And the interesting thing is when we looked at microbial diversity over time, there was definitely a shift away from certain taxa and favoring other taxa. So again, showing that we were able showing that the host genotype is selecting the endophytic community in which it wants to interact. By way of summary, we've characterized the bacterial community in the native environment. We've isolated thousands of isolates. We've taken these isolates and we've characterized them in terms of their functional capacity as well as their genetic diversity. We're in the process of reconstructing communities. Those reconstructed communities are directed towards testing this idea of a lottery hypothesis, that individual taxa aren't necessarily the key to determining favorable or negative effects on the performance of the host, but rather which functions those bacteria or fungi bring to the relationship. These are the things that we need to do better. We need to move beyond OTU and 16S pyrosequencing for genotyping. There's really not much to be learned there other than phylogenetic position. It really doesn't give us predictive power in terms of function at both the bacterial or fungal level. We need better implant imaging techniques. We need to leverage the new long-read sequencing technologies so we can get more definitive alignments when you resequence a community, both at the genome level and at the transcriptome level. We need better single-cell capturing and culturing techniques. A lot of the bacteria that we have in culture were those that were easily cultured, of course. And the slow-growing bacteria, slow-growing fungi are underrepresented, and there's certainly a large segment of the community that we couldn't capture or culture. So we need better single-cell capture and culture techniques. And we need to apply sophisticated statistical approaches to metagenomics and metatranscriptomics, metaproteomics, to better characterize and understand how these communities are interacting. With that, again, I'd like to thank the sponsors, our collaborators at Duke, ENRA in France, the JGI for all the sequencing, the University of Washington, and Oak Ridge National Laboratory. Thank you.
Thank you for a very, very nice talk. That was wonderful. Tons of information. Actually, I have a question about, it seems like there's an analogy between what goes on with the control of pests or pathogens in plants, and the control of pathogens in animals. And I'm wondering if anyone's studying the effect of the use of pesticides and herbicides on the microbiome that's associated with plants. And the second question I would have is anyone studying the animals in the rhizosphere? The nematodes, the small insects, and so on and so forth. Seems to me that passage through their guts would have a tremendous impact on the ratios and the diversity in them.
To the best of my knowledge, the fauna of the soil is not being studied in very much detail. It certainly plays a critical role in carbon cycling and the movement of bacteria across soils. Your first question was pesticide applications. We work mostly in native environments. In agronomic context, most of the disease resistance occurs at the genetics level of the host. So for wheat rust, for example, they'll select resistant lines of wheat. So pesticides are used, but not as commonly as genetic selection. Question is still relevant there. If we select for resistance, let's say for leaf rust or melamporous leaf spot wilt, does it influence the microbiome? We're beginning to answer that or ask that question in the common garden. So we scored the common garden for foliar pathogens, and then trying to correlate the occurrence of resistance at the foliar level to pathogens and the diversity or abundance below ground. We don't have an answer to that yet.
Becky? Yes, very interesting, and I'm a plant pathologist, so I'm very interested in what you had to say about the populations in the soil. Actually, a couple of questions. One is, did you say that the populace are clonally reproducing and that they that one whole area was one genotype?
Yeah.
So how does that impact their ability or their propensity to select in this lottery hypothesis versus carrying their populations with them?
It's a great question. Poplar has an interesting habit of clonally reproducing, so that was a single genotype. So a single host can explore acres and acres of soil, accumulate microbes that are beneficial over long periods of time, and distribute those through the conductive tissues. So that's very, very possible. The second way that poplar establishes itself clonally is that in the fall, typically we see yellow leaves and we take pictures of this grand landscape. In addition to shedding leaves, they also shed small lateral branches that then float downstream and then bed in the soil and propagate themselves. So in your biology classes, never say that plants don't move, because poplar actually intentionally casts itself into the environment to recolonize unexplored niches. It brings with it then its microbiome when it does that. And so it carries with it part of its original bacterial components, but it recruits new bacteria in new environments based on soil pH, moisture availability, and so on.
Thank you. We're going to hold off further questions and discussion to the end of the session. We'll bring Dr. Tuskan back.